Welcome all to this second webinar from NCOH, the Netherlands Center for One Health. And today's topic will be aerosol, and the webinar is entitled Aerosol, the Complexity of Science. Aerosols are very important. Actually, they are to be considered hot in the discussion on how that we control transmission of SARS-Cov-2 in our country and actually globally. And it's either to be said that you're either against or you are for aerosols. Well, today we have three speakers who will tell us about the science of aerosols. And I will introduce them to you before we start. And I have a few housekeeping announcements. First of all, this webinar will be recorded for your information. Secondly, you have the possibility to chat. You can chat with each other, related or unrelated to the presentations on the right side of your screen. But if you want to bring in a question for the Q&A session at the end of the webinar, please fill in your question at the top of your screen. The questions will be collected, and in the end, we will distribute the questions to the three presenters. I think we have a very interesting lineup of speakers for this webinar. The last speaker will be Dr. Karen Ellen Veldkamp, Head of Infection Control at the Leiden University Medical Center, who will talk about the prevention of SARS-CoV transmission by aerosols in hospital settings. Second speaker will be Dr. Litwin Smit from the University of Utrecht, who will give us her experience of measuring SARS-CoV-2 in the air, with her experience from studies in mink farms and a slaughterhouse. And we will start with our first speaker, that is Sander Herfst from the Virus Science Department at Erasmus. And his subject will be the collection of infectious aerosolized respiratory viruses from the air. I invite Sander to start his presentation.
All right, so thank you, Mark, for this introduction, and thanks to the NCOH for giving me the opportunity to talk about our work. So even today, we only have very little knowledge about the transmission routes and the transmission kinetics of respiratory viruses. And the knowledge that we have is mainly based on animal transmission experiments, human challenge infection studies, outbreak reports, and household studies with only very little quantitative data available. So to illustrate that there's still a lot of debate or actually confusion about the transmission routes of respiratory viruses, we previously summarized the isolation guidelines for respiratory virus infections from different national and international organizations, national, European, American, and international. And we compared that to the experimental evidence on transmission routes. And that's seen on the x-axis. On the y-axis, it's indicated if those guidelines aim to prevent transmission via direct or indirect contact, transmission via large droplets that don't stay in the air very long and that quickly settle on surfaces, or via small aerosols that can remain in the air for a long time. If we then look at three very well-known respiratory viruses, so influenza virus, rhinoviruses, and human coronaviruses, indicated by the red squares, then we can see that all the symbols in these panels are scattered, meaning that there's no guidelines at all, or that there's no agreement on the transmission routes, likely then due to the limited experimental evidence. So to better understand the likelihood that a certain virus can be transmitted via the air between humans, quantitative data is strongly needed. So first, we know on the left side that how much infectious virus is shed in the air by an individual, and also what the duration of virus shedding is. Then secondly, we need to know how much infectious virus is shed in the air by an individual. Second, we need to know how stable the virus is and how long the virus remains infectious in the air. Thirdly, on the right side, we need to know the infectious dose of the virus, is the number of infectious virus particles that are needed to infect a new individual. And information on the infectious dose of a virus can come from humans or from human or animal challenge infection studies. Those have been performed previously for influenza viruses, and from those studies we know that the ID50 for influenza is only 5 virus particles. It means that if you would infect a group of people with 5 virus particles, then 50% of those people would in the end be infected with the virus. But for the virus shedding and the stability in the air sampling can help. And air sampling is the collection of viable, so infectious respiratory virus from the air. And it can help us to better quantify the virus shedding and the virus stability in the air. So I think in general that we all agree that influenza viruses can be transmitted via the air. So if there would be another virus which, for example, would be shed in the air 100-fold less, and that would be less stable in the air and lose its infectivity, or that needs a much more higher infectious dose, maybe 500 particles instead of 5, transmission by the air is for this virus probably less important. So to better understand transmission via the air, we started to do experiments with air samplers. So the first one that I would like to introduce is the biosampler or the impinger, and this method is based on liquid impingement. So air comes in at the top, and the aerosols end up in one of the 3 nozzles where they get accelerated at the bottom. Particles leave the nozzles at a very high velocity, and then they get impinged into swirling liquid collection medium. The second one on the left-hand side is the Anderson cascade impactor. The version that we use consists of 6 stages, and each of them containing a petri dish with collection medium. In our experiments, we use semi-solid gelatin. I will not go into the technical details, but a big advantage of this sampler is that the sampler size fractionates the particles, with the larger droplets ending up in the first stage and the smaller aerosols ending up in the last stage. So you can actually say something about the size of the droplets and the aerosols that contain virus. As a third option, we use the in-house developed electrostatic precipitator. So this consists of a glass chamber, and this contains a corona wire at the top that produces positive ions. So when the aerosols enter the glass chamber from the left side, they will get positively charged, and they are attracted by the neutral collection medium. So these 3 different samplers we use in an in vitro setup to collect to compare the collection efficiency of the 3 samplers. So this is our setup that we use. So in the class 2 cabinet, we have on the left-hand side, we have a nebulizer that we use to aerosolize viruses. Then we have a Nalgene box in which the viruses are aerosolized, and to this box we connect either one of the air samplers. These air samplers are subsequently connected to vacuum pump to draw the air through the air sampler so we can collect the viruses from the air. And we did it with the 3 different air samplers that I just presented. So we have the electrostatic precipitator that we also call the Art Vector, named after the guy who designed it. His name is Art. Then we have the impinger and the impactor. And we see for 2 different viruses, so H1N1 influenza virus and for the human metapneumovirus on the right, that if you compare the amount of collected infectious virus in red with the amount of aerosolized virus in blue, that the impinger and the impactor actually worked very well. So almost all the virus that we aerosolized in the chamber, we were able to collect it with the impinger and the impactor. Unfortunately, the Art Vector, so the electrostatic precipitator, did not work as efficiently. So a lot of infectious virus was lost there. So the collection of the infectious virus using the impinger and the impactor was very efficient, as I just showed you in our in vitro setup. But unfortunately, these samplers have a detection limit of 500 to 600 nanometers. And you can imagine that a 100 nanometer influenza virus may still fit in a 300, 400, 500 nanometer aerosol. And we could miss those aerosols in our system. So that's why we wanted to improve the collection of these submicron particles based on the collisional growth method, and it's demonstrated here. So this basically means that aerosols are actively enlarged before they get collected in the air sampler. So as illustrated here, if you mix aerosolized virus with vaporized water in a mixing chamber, then the particles collide and they merge into bigger aerosols. And these enlarged aerosols they can subsequently be collected more easily. And this collisional growth method, we evaluated it in a different in vitro setup that is shown here on this slide. So on the right-hand side, let me show it. So on the right-hand side, you see here a box that is filled with water with PBS, and it contains the ultrasonic mist makers that are normally used in humidifiers. So here we produce the mist, and this mist from this box was then blown with a little pump into a 3D printed mixing chamber, where it was mixed with the air that contained the aerosolized viruses. And in this experiment, the aerosolized viruses came from a circular tube in which virus was aerosolized here and was circulated in the circular tube by this ventilator that is built in. So in this way, we can circulate viruses for different lengths of time, and we can actually study how this affects the virus stability and infectivity. And using this setup with a mist maker and a mixing chamber, we could indeed improve the collection of infectious virus using the air samplers. So that's all in vitro, of course. So we were also curious to see if we could use these samplers in an in vivo setup to see if we could collect infectious virus, of course. So this is our ferret transmission setup that we've used previously to study transmission of several influenza viruses, but also for coronaviruses recently. So it consists of a donor animal on the right side and a recipient animal on the left side. And the donor animal is inoculated with the virus of interest, and then next day recipient animal is added to the other side of the cage. The animals are separated by 2 steel grids, so they can't touch each other. So the only way that the recipient virus can be infected by the donor, the recipient ferret can be infected by the donor ferret is via transmission via the air. And this is how we usually do our experiments, but in this typical experiment, we added air samplers to the system. So in this case, the air that went from the donor ferret to the recipient ferret, and then via tubing, it went into a big mixing chambers indicated here in yellow. And to this mixing chamber on the left side, we see a mist maker that we generated with PBS this time. And we see the 3 pumps and 3 impingers in which we collected viruses from the air. And the medium, so there's 20 milliliter of virus transport medium in these samplers. And they were actually, every 2 hours they were collected and replaced with new medium for 5 days. And of course, we worked with viruses here, so everything needed to be contained. So this whole experiment took place in a class 3 isolator in our high containment facilities. So if we then look at the results of the transmission experiments, the ferrets did exactly what they needed to do. So this graph shows the infectious virus dose that we see in the donor ferrets, indicated here by these 2 lines. But also the infectious titer in the recipient animals. And you can see that after 1 day, there's already a little bit of virus in the recipient animal, and at 2 days, there's quite high infectious titers. So the virus very quickly jumped from the donor ferret to the recipient ferret, resulting in positive throat and nose swabs in the recipient animals. So that part of the experiment worked. So there was indeed virus transmitted from the donor to the recipient. But let's then have a look at the air samples. So each bar shows the amount of viral RNA that was collected during a 2-hour period. So every 2 hours we replaced the medium with fresh medium. So we did quantitative PCR. You see a small bar here in the beginning in the first hour. That's probably right after the inoculation. Then after 8 hours, which is approximately 1 replication cycle of influenza, we see some virus. And then after 16 hours, we see continuously in all the 2-hour samples that we collected, we see a lot of viral RNA present by TACMAN. And it's quite a lot of RNA actually, but unfortunately we were not able to culture viruses from these samples. So although the collection method worked, it was still too tough or too hard for the virus particles, so they probably did not survive the collection method, or they didn't survive the traveling through the air to the sampler. For conclusion then, we show a high collection efficiency of infectious virus with our in-house developed impinger-based collisional growth method sampler in vitro, like I showed. In the ferret model, unfortunately, no infectious virus was collected, but viral RNA was collected continuously from 16 hours post-infection of the donor onwards, which shows that it's really difficult actually to collect infectious virus from the air. So therefore, more elegant methods should be developed or applied to ensure survival of collected viruses. And we are now exploring a condensation-based sampler in our labs. And just to finalize, knowledge on the combination of virus shedding kinetics, stability in the air, and infectious dose will help to determine the relative contribution of transmission via the air to transmission of a respiratory virus. And with that, I would like to thank Jasmin Kutter, Dennis de Mulder, Theo Westerboer. They did most of the work. Thank you.
Thank you very much, Sander.
I would like to give the word back to Mark.
Yeah, already had the word. Thank you very much, Sander, for this nice explanation of the complexity of just measuring the virus in the air. One very short question: could you speculate, or do you dare to speculate on the let's say on the role of the aerosols based on these single experiments in which you could measure RNA but not living virus?
Yeah, so that experiment was for us a little bit of a disappointment, of course, because we've shown that the viruses can travel through the air, and in fact, a naive ferret over a short distance. And we know that influenza viruses, that it can be transmitted via the air. But so it was a bit of a disappointment that we could not collect recover infectious virus from the sample. So that really means that we should really improve the methods that we have at the moment. That answers your question.
Yeah, thank you very much. So for more questions, we hope to get questions from the audience. You can point these and then report these questions on top of your screen. And in the end, we will have a Q&A with all the three speakers. So then we move to the second presentation, which will be given by Lidwien Smit from the University of Utrecht. From ferrets, we go to minks and then to a slaughterhouse. And she will tell us her experience on, let's say, measuring SARS-CoV in the air. Lidwien, the floor is yours.
Yeah, thank you, Mark, for the introduction. Yes, I will talk about first mink farms and then about our experience in a slaughterhouse. So the first NCOH webinar was completely dedicated to infections in mink farms. So I'll just give a very brief summary of the current situation. Well, infections of SARS-CoV-2 were first detected in the Netherlands at the end of April, then followed also by infections in farms in Denmark, Spain, and the United States. In the Netherlands, we found that introductions from humans were the most likely route of transmission. We've seen widespread mink-to-mink transmissions in farms, also mink-to-cat transmission, and mink-to-human transmission that was most likely. Yesterday, a manuscript was uploaded on bioRxiv, so I invite you to read this if you're interested in seeing the research about the sequences that have been written by the colleagues in Rotterdam. In total, 68% of tested workers were positive. So that's quite a high frequency of positive tests in this working population. And until now, 44 out of the 128 mink farms in the Netherlands were infected. And well, all the farms that are found to be infected are immediately culled. The minister also announced an accelerated ban on mink farming last week. Mink farming would be illegal in the Netherlands as of 2024, and now that will be March 2021. And that's because mink farms pose a potential risk for public health, because it's forming a reservoir of the virus that is impossible to contain, it seems. Well, one question that we wanted to answer is whether SARS-CoV infections in minks are also an environmental health risk. And when the first farms were detected, as a precautionary measure, roads around the farms, so in a 400-meter radius, were closed for pedestrians, also for cyclists. And we aim to study whether you could find any viral contamination, both in the mink farms, but also in the surroundings. We did it by active air sampling. We collected inhalable dust, which is a relatively coarse dust fraction, and also by PM10, which is a PM particulate matter less than 10 micrometer fraction. We collected the dust on Teflon filters, and we conducted both 6-hour sampling, but also sampling over multiple days. And the filters were extracted and analyzed by qPCR in Lelystad. Well, the results of the stationary and personal air samples in the mink houses. So on the right hand, you can see a stationary air sampler in the mink house, and on the other picture you can see 2 field workers who are carrying the air sampling equipment with them. And we detected viral RNA both in inhalable dust and sometimes in PM10, but mainly in the inhalable dust samples. And levels were higher in the personal air samples because people are moving around in the stables, and the stationary samples are just put in one spot. So you have to be a little bit lucky to find the good spot where you can find air samples with viral levels above the limit of detection. We also found in a few samples that air sample virus sequences that they cluster together with the mink sample sequences from the same farm. So that's not entirely surprising, of course, but it was nice to see that. Apart from air sampling, we also did some additional sampling. We took, for instance, swabs of cages, both from cages where animals were housed that were recently deceased, but also from live animals. We took samples of bedding material. We also conducted passive air sampling, and that's by just simply laying a piece of electrostatic cloth for a week in the farm. We also took feces samples, and we took swabs of drinking water supplies, and we took some feed leftover samples. And as you can see, the number of detectable samples is very high, especially from the swabs of the cages, from the bedding material, and also from the passive air samples. We looked at the determinants of viral load in those samples. Some had a really very high viral concentrations with CT values around 20. And we found that, well, the farm was a determinant. Some farms had higher levels than others. Sampling in cages with recently deceased animals had higher viral loads than those with live animals. And a week in the study was also a determinant. We saw a decline over time of the viral loads. Very interestingly was also that 2 weeks after culling, we went back to one of the farms, and we still found many samples to have detectable RNA in, well, both in bedding material, but also in the EDC passive air samples. Beyond the farm premises, we did not find any measurable SARS-CoV-2 RNA. So both in inhalable dust or in PM10 samples, we found a few measurable samples really on the farm premises, so really close to the entrance of the mink houses. But as soon as you came out of the fence of the farm, we could not detect any RNA in the air samples. We did both upwind and downwind gradient measurements, so we stood very close to the farms and then took into account the wind direction. We also did long-term sampling, both on the property but also in the village close to the mink farms, and we also included a background location really outside of a mink farming area, but we did not see any detectable samples there. So because of these analysis, the precautionary area measures were lifted because it was deemed safe to be around the mink farms. So in conclusion, the occupational exposure to high levels of SARS-CoV-2 RNA is found in airborne dust in mink farms. However, the viability of this virus is unknown. We did not test for viability. We tried to culture a few of those environmental samples, which was not successful. the mode of transmission from the minks to the minks. So the infection went through the whole farm, but it's unknown whether the minks got infected through humans or through handling the mink. And also the mink to human infection, the mode of transmission is unknown. Whether the airborne dust plays a role, whether there's airborne transmission, we really don't know based on those data. The risk of environmental exposure appears to be negligible. That's also confirmed by looking at viral sequences in people who had COVID-19 in a neighborhood. We test samples of people living close by, and they all had different viral sequences than the mink farm sequences. So apart from an occupational risk, it doesn't seem to be really an environmental or public health risk. Then a very different topic, but also in an occupational situation with a large number of clusters worldwide, is in cool working areas, for instance, slaughterhouses or meat packing industries. And there is many questions about those working areas. For example, does the role of temperature, is it really the cool working area, or is it relative humidity, is it air recirculation, or is it simply because people shout, they're close to each other? Those working areas are characterized by a lot of migrant workers. They share housing, they also share commuting. So it may also simply be that they're very close to each other, and clusters are because of the close vicinity to each other. We did some preliminary sampling in a cool area of one Dutch slaughterhouse that had a COVID-19 cluster. And when we looked at surface swabs, we found 6 positive swabs in total. On the picture, you can see the 6 guilty surfaces. So as you can see, it's mainly high-touch areas. And we had 1 positive personal air sample, and that was of a worker who also had a positive test for coronavirus himself. We also took stationary air samples. We inspected the ventilation system filters and looked for viral RNA, but they were all negative. The problem with this kind of environments is that it's very difficult to conduct field work. You're often too late to party because the humans are the source themselves in this case. Well, often preventive measures are then taken when a cluster is identified, for instance, by triage or cleaning and disinfection. So a lot of questions remain, but it's very hard to conduct studies in this kind of working areas. So I'd like to thank you for your attention and also thank all the people who were involved. It's a lot of people, I cannot mention all of them. Almina de Rooij from Utrecht University conducted a lot of the work presented here. And I would also like to thank Arjan Stegeman, Wim van der Poel, Marion Koopmans, and many others, also from the Netherlands Center of One Health. So thank you very much for your attention.
Thank you very much, Lidwin. Short question to bridge to the next presentation. Can you speculate on how the mink farms got infected? If the viruses that were found in the minks did not resemble the viruses that were found in the people surrounding the minks, is it still likely that it was introduced by humans?
Yes, that's the most likely option. Well, we discussed all the other possibilities, for instance, wildlife, airborne transmission, but they are all highly unlikely because of, well, sometimes you see 2 mink farms that are really next to each other and they have a completely different viral sequence. The mink sequences are clustering together with the worker sequences. So it's possibly multiple introductions by workers who come to the same farms, and it's very difficult to get a good idea of who is working where and who has contact with whom in this. And there's also a lot of migrant workers from Poland, and it's not entirely clear who is going to which farm, when, etc. So that's still the most likely explanation.
Thank you. So for attendants that are watching the presentations and have questions, please report your question on top of your screen, not in the chat box, but on top of your screen. And then we will address these questions in the Q&A after the next presentation, which will be the third presentation by Karen Ellen Veldkamp from Leiden University Medical Center. She's head of infection prevention there. And she will tell us about prevention of SARS-CoV-2 transmission by aerosols in hospitals. Karen Ellen.
Okay, thank you, Mark. I will tell how we prevent in the hospital the transmission of SARS-CoV-2, especially in patients who are admitted to the hospital, and what will be the role of prevention of aerosols. So if we look at the transmission of SARS-CoV-2, then we are mainly focusing on the prevention of the droplets, the spread by droplets, when people are shedding droplets from their mouth or throat, and they will be inhaled to a person which will get infected, but also the droplets of course can fall on surfaces and objects, and if you touch those surfaces and objects, you can pass these viruses with your hands to your mouth or eyes or nose. So the main transmission is by droplets and contacts, but of course it's important to know what else will be possible. Okay, so well, I think this is no news for anybody, the transmission by droplets or contact. And then what will be the risk of infection after close contact with a COVID positive person? So if we have an infected person who is shedding virus particles, when will you become infected? And that depends on the duration of your contact. It depends, of course, on the frequency, how often do we have contact with this person, but it also depends on the viral load which is shed, but also which is inhaled by the vulnerable host. That's the chance to get infected, and then of course it will not be certain if you will get the disease, and the disease will depend on what is the immunity of the host, and of course how many virus particle are you infected with. So if you get really the disease depends on different factors. The presence of viral RNA cannot be directly interpreted as potential infectious virus, as was shown by Wolf in 9 patients with mild COVID. He showed that in sputum and also in throat swabs, after 8 days, there was still viable virus culturable, but there were negative cultures, although there were positive PCR results from till 14 days in these patients. So of course, when we are talking about infection control, it's very important to know when people are not infectious anymore. Another study from Kampen from Erasmus University, when we are talking about definition of an aerosol or airborne or a droplet infection, then we are talking about aerosols. There are 2 definitions you can use. You can use the cloud of droplets of different size, with big droplets and small droplets and nuclei, but often they talk about aerosols when they're talking about small droplets, which are smaller than 5 micrometer, and the nuclei. And of course, if you're talking about infection and infection prevention, you have to know if these aerosols are infectious aerosols. And we're talking about infectious aerosol if the small droplets or nuclear are carrying viable microorganisms. Airborne infections definitions are infections caused by these small infectious droplets or nuclei. And then you can think of tuberculosis or measles, for example. And droplet infections are the infections which are caused by microorganisms, which are mainly spread by droplets which are bigger than 5 micrometer. So the difference between droplet and airborne transmission is, of course, the precautions you have to take. And the airborne transmission, then the tiny particles which are possibly produced are suspended in the air, and they travel for a longer period, but also further. So that's what you have to take into account if you're talking about precautions. In the hospital, often infectious aerosol generating procedures are performed, and these medical procedures which are generating infectious aerosols, these depend on the procedure. Is it aerosol generating? It depends, of course, if the person where you are performing the procedure is having an infection, and it depends on the localization of the infection and, of course, the localization of where the aerosol generation procedure is done. So in the Netherlands, we made a guideline because there were a lot of questions which procedures would be potential infectious, especially for SARS-CoV-2. So then, of course, we looked at what sites the infectious viruses can be found. And of course, also the World Health Organization depicted several aerosol generating procedures which are dangerous to perform without the right equipment and the right protection. So these are the open suctioning of airways, the resuscitation, and of course all kinds of invasive ventilation. The precautions, they depend on the risk of transmission, and then we have to think of personal protective equipment, which healthcare workers have to wear, and of course also how we isolate the patient. And if we are talking about the personal protective equipment, then the protection of the respiratory tract is by a mask, and then it depends on if we are talking about a contact droplet infection, then you have to carry a surgical or medical mask. And if you're performing aerosol generating procedures or treatment, then you have to wear an FFP2 respirator. The protection of the eyes is by safety glasses, and if aerosol generating procedures are performed, goggles are used, and then a gown with long sleeves and gloves. And which is most important is, of course, that all the protective equipment is properly worn, but also that the doffing is very important so that you don't contaminate yourself when you are putting off the protective personal equipment. Of course, on the other hand, we have the isolation. We want to separate COVID positive and suspected patients from the other patients and from healthcare workers by isolation, and therefore we use a single room. But if aerosol generating procedures or treatment is performed, we use isolation rooms. And if we look at source isolation, so the special rooms, then we see that in the hallway, we see that there is a normal pressure, but that there is a negative pressure in the entry room, which is used as a lock between the patient room and the hallway. So the airflow will go from the hallway to the lock and from the lock to the patient room, so that no air can escape from the patient room and go into the hallway. This is important, especially when aerosol generating procedures are done, but not if we are talking about droplet and contact spreading, because then, of course, you don't need an isolation room because the droplets don't carry that far. If there is a shortness of isolation room, you can think of cohortation, and this means that you have several rooms with a separate bed, which are separated from each other, but there is just the healthcare workers will carry on this cohort. They will carry all the time their personal protective equipment, so they can go from the one room to the other room. And this we can only do, of course, if the patients are really proven COVID positive and not if they are only suspected. There are several studies of air and surface contamination by SARS-CoV-2. This is a study in a tertiary hospital in Wuhan. They looked at several patients with mild and severe disease, and also they grouped them by seroconversion and by the positivity of their PCR tests. And the result was that in a total 367 air and surface swab samples were collected from the patient care areas, where 15 patients with mild disease were treated and 9 patients with severe or critical disease. Only one air sample taken, which was taken during the intubation procedure, tested positive, and it was so that mostly the high-touch surfaces were slightly more likely to be contaminated with SARS than the low-touch surfaces. So if you think of contact transmission, this is of course very clear. Another study looked at aerosol and surface distribution in 2 different hospital wards. One was an ICU. In this ICU, that was a large floor space with 15 open cubicles, and you had a central open area where the healthcare workers were staying. And then you had a general ward, and it was in the picture B, and these general ward was all separate, so no isolation rooms, but all separate rooms with closed doors. And it was found that the contamination was greater in the intensive care units than in the general wards, and that the virus was widely distributed on floors, computer mice, trash cans, and the sickbed handrails. So again, the high-touch areas and was on the ICU was detected more than 4 meters from the patients. And if you look at the right side, you can see that the rate of positivity at the ICU was 40% where the beds were, and in the central area 12%. And if we look at the places on the general wards, you see a rate of positivity in the hallway of 0%, and in the beds in the patient rooms of 12%. So to summarize, the droplet precautions we use for COVID positive patients are effective for general wards. Airborne precautions should be taken when aerosol generating procedures or treatments are performed. And more and further contamination of the environment is found on the ICU wards where patients with severe disease are treated, and also where more aerosol generating procedures and treatments take place. It's important to keep in mind that the presence of the positive PCR is not equal to potential infectious virus if we are talking about the measures we have to take. Thank you.
Thank you, Karen Ellen, and thank you, Thomas, for helping in presenting the slides so that they were more totally presented, fully presented at the second part of the presentation. So, Karen Ellen, very short question. If these aerosols, let's say, if the measures taken for contact isolation were effective in the hospitals, do you think that aerosols could be important outside hospitals?
Well, I think that the main route of transmission is by droplets and contact. And of course, there is a lot of literature about the spreading of aerosols in specific situations, like in koren and kerken. But to answer your question, I think that it's maybe in specific situations where people are really having high viral loads, screaming in close distance, that people can get infected. But actually, I think that still then the main route is by droplets, because all those articles are really talking about people who are close together and frequent together, and maybe having a social network where they see each other quite often and also do other things together.
Yeah.
So, of course, it can't be excluded that with singing and yelling, little droplets. Of course, little droplets are performed when you're coughing, when you're sneezing, but I don't think that there's much difference between singing and coughing and sneezing.
Good, thank you. Lots of questions have been submitted, so also since we have 5 more minutes, but my closure will take only a few seconds. So many questions for Litwin for the second presentation. So I will just drop some. One of the questions is, how come the drinking water supply was positive in the mink farms? The person that answered the question presumes the water supply is a closed system. So how can it then happen that the water supply, the drinking water supply, was positive for the virus?
I think that's a very simple answer. Both the drinking water supply and the feed residues were most likely positive because of contact with the animal. I mean, they touch it with their snout, and so that's most likely. It's not that the drinking water itself or the feed is contaminated.
Okay. Another then maybe easy question, at least to answer, would be then is, wouldn't airborne transmission in the mink farms be evident from the geographic location where, let's say, the first or the majority of the infections were detected?
No, absolutely not. Well, one of the things is because the viral sequences can be very different, even with farms that are located next to each other. And so we don't see any relationship between geographical distance and the viral sequences that are detected. The fact that it's located in one area is most likely because many mink farms have the same owner. They have workers who come to similar or to different farms. And also because this area was initially one of the most affected areas. So perhaps the first introductions were, well, because of the owner, for example, got infected during Carnival and then infected the animals and also the co-workers, and then it spread in this area.
Okay, thank you. A question for Sander. Is the in-house developed impringer-based sample collection study published? Is a question.
No, it's not published yet. So most of the data I showed was actually not published yet. I'm not sure when we're going to do it anytime soon. But yeah, if you want to have more information about the system, we are always happy to collaborate and to share our findings. So I don't know who it is, but just shoot me an email. I can be found on the internet, or just s.herfst@erasmusmc.nl, and I will be happy to share any information and knowledge that we have.
Thank you. A question for Karen Ellen. Someone, I'm not mentioning any names here, says, I'm wondering why aerosols are defined to be less than 5 micron droplets, since aerosols between 5 and 10 may well stay airborne too. So where does the cutoff of 5 come from? And maybe that's also a question that Sander or Litwin can have an answer to. But it was initially meant to be answered by, at least to be asked to Karen Ellen. Karen Ellen, you're muted. You must unmute.
Like I told in my presentation, yes, what do you call an aerosol? I think aerosols are just a big cloud with all kinds of droplets from different sizes, from very big to very small. And if we are talking about aerosols from 5 micrometer, I don't know exactly where that comes from. No.
Any of the other 2 aerosol experts?
Well, I think it's a general assumption that people use a lot, but there's not a very clear cutoff. Some people suggest that also anything up to 100 micrometers should be an aerosol, because also these particles that are much larger can remain in the air for a prolonged period of time. So there's it depends on who you talk to. You get different answers there, I would say.
But I think there's a difference between aerosols and airborne.
Right, there I would say that airborne is just a combination of transmission either via aerosols or via droplets, right? So that is anything that can be expelled into the air, remain infectious while being in the air, and can infect another human being. That's all called airborne, but airborne doesn't make a distinction in the size of the particles.
Yeah, in infection control, it's a big difference. Because if it's airborne, you really have to take into account that you use isolation room, that you take other protective equipment. So in the hospitals, we make the difference between airborne and droplet depending on how it's transmitted, how far and how it really will be transmitted by air.
So this confirms very nicely how I started, that there's a lot of debate still there about what is what and what sizes does it concern.
Yeah. I think a final question then, we come back to Litwin. Let me see the question here. What's as minks farms, and I'm probably you need now to take the position of a politician, but as the minks are no environmental risk for the populations and minks are culled anyway each year, is culling then still needed if a mink farm is found to be positive?
Well, I think it's a good idea to do culling because we are, of course, a very densely populated country. In Denmark, for example, they don't cull anymore. They did it at first and now they don't. I think there's still the risk of infection of the workers or other people visiting farms or visiting people at the farms. Especially now, currently there are very few human cases in those areas. You don't want to have new clusters. So I think culling is right now the best solution. Although from a public health perspective, you may argue that it's probably not necessary, but yeah, that's maybe also a political issue, I agree.
Yes, I don't think we should have the Kamerdebat here.
No, we shouldn't.
Also not because, let's say, we're running out of time. We have come to the end of this webinar. Please rate the webinar, how you find. I think that should be somewhere on top of your screen where you can fill in the evaluation form of this webinar. All presentations will be placed on the NCOH website. And I think with thanking all the three presenters and mentioning that the next webinar will be on October 1st, I don't see any topic yet, so that is still to be decided. With that, I would like to thank all presenters and the audience for attending this webinar and hoping to see you again with the next webinar from NCOH. Thank you very much. Bye.